Time. I also have good news. I marked your unknown assignments. Took me a bit of time, but if you want to also have a look at your slides, they're also in the middle bench. I'll briefly share with you all, and you'll likely be working as groups for majority of today's lab, minus the street plating exercise one. But I'll show you pretty much what you need, hopefully in due time. I know some of you just came in, so just please bear in mind that you also pay attention. And if you have questions or if you haven't shown me your notebook, you could do so near the end of today's lab or when we have breaks. Um, there was something I wanted to say. Yes. So in terms of figures, I just want to reiterate this for your lab notebooks. Please make them as descriptive as possible. I know some of you have just labeled micrographs with essentially pictures of your bacterium, but you need a label. Okay, this is the bacterium, the name that you're assuming, right, in the unknown bacterial assay for your gram test or your gram staining test or your endospore, etc. And just make sure that you're as descriptive as possible, such that if you were to present this to someone else, be it from our lab or outside of our lab, that they should be able to know what exactly you were studying. So just bear that in mind, everyone. Now, let's go straight ahead. We'll be going through the different slides and talking about what today's exercise or exercises will include briefly. Okay, so starting off, right, we have, and I mentioned this earlier, streak plate method, right? We're going to differentiate this from the spread plate method we did last week. Since essentially that'll be what we're starting off first for today. Uniquely for the plaque assay, you'll be working as groups of 4, but this importantly will be for your first formal lab report due on March 1st. So just have a look on Quirkus, the module section or assignment section to see more details. But you'll also be referring back to your results for this week or the methods that you do for this week to follow in the following week. Thereafter, bacterial transformation comes as our third exercise, and the fourth, sorry, give me just one moment. Yeah, so following the third exercise, the bacterial transformation, we have our fourth as conjugation. Remaining the same set are 3 to 4, depending on the numbers. But essentially, you'll be working and making sure that you label all the instruments that you use for today. Also bear in mind that the materials that you use are specific for each exercise, some of which are on your benches, some of which are on the middle bench. For example, transformation, we have that apparatus in here, the broth, the transformed E. coli, HB11 statement, and then we have Aluteus here for your sheet plating and NA plates that we'll be using shortly. In due time, the lab technician is going to be adding in, I think he has he's going to add in liquid the broth for culture at 55 degrees, so just bear that in mind for timing. We'll likely go with conjugation before transformation, so we'll just adjust schedule accordingly. But nonetheless, you know that for exercise 5, it's actually going to be the upcoming slide, but essentially it's what you would learn from the appendix or from your lab manual, exercise 5. Exercise 5, sorry. And then serial dilutions, I believe Dr. Brunt said that she placed them on your benches. Is that correct? Yeah. So you can also have a look. Well, when we have time, you'll be able to record your observations. I would just say in the meantime, focus on the apparatus or the exercises you have to work on today. And then as groups, or actually as individuals, one of you can start counting the colonies when there's enough time. So that way, because there's about 3 plates each group, each of you could at least do one for counting. And then we'll talk briefly about how you would multiply to get your CFUs, sorry, your, yes, colony forming units. Any questions in the meantime? Or to get your original density is what is normally called. Now, media preparation, that's what I was just talking about, exercise 5 right here. That's where you're going to be just looking back here to Appendix D rather, and E, to learn more information. But it's pretty much what we've done, what we've used from the past, and now in terms of preparing those medium themselves, which is actually quite interesting and impactful to knowing more about how you would be able to also prepare your own media down the road in the future. Now, for plating, exercise 1, streak plating, right? This is the question that you're asked in bold or referenced in bold in the lab manual. Let me ask, what's a pure culture? Or does anyone know what the difference is between a mixed and pure culture? Okay, perfect. Were you about to say the same or anything else? Yeah, okay. So, both of you are correct in that note. Essentially, a pure culture refers to a culture like the plates that we have, or even on the NA slants that only have one colony, generally one species of a genus. So it could be Amluteus, for example, whereas a mixed culture can have multiple different types of bacterium for different genuses or geni. So just bear that in mind because that also helps us knowing how we're going to actually, eventually create our own or develop pure cultures like the one that's referred to here. So pretty much the idea is actually it's quite explanatory, and you'll probably also see this in your lab manual, and hopefully you've drawn it out in your flowchart for exercise 1. The idea is that you're essentially streaking out 1/3 as if you're making let's see. Essentially, it's as if you're making a T, and there are different ways to do this, but one way that's commonly shown in the lab manual, sorry, is essentially that T, essentially, where you essentially start off with your amluteus shown on the red tray down there, pretty much so that you all know. Each group will take one plate. If in case members of this group don't come, if you mind again, the idea is that essentially as groups you'll be completing this task for about 15 minutes, and it's very simple. Just essentially you taking just what you've done before, but a greater amount of luteus from your NA slants, and then you're just streaking like this, pretty much how they've done it, except once you're done that third portion, you turn around, right? Like you turn a third way, just showing an arrow for illustration. You turn around your agar plate, following aseptic technique as always, and then you spread or streak from the previous region and just keep going up and down, up and down, up and down until essentially you repeat that process for the second third essentially or second portion or quadrant of your agar plate, and then lastly you just return, like you repeat that process with another arrow, for example here, and then you continue that streaking pattern such that eventually, like before, you're actually diluting the amount of bacterium. And does anyone know why that would be the case? Like, why would you see only, like, as you see here, it's going like to a large amount of bacterium that you see in many colonies smeared together until it eventually reaches like very few dots in this location. Does anyone know why it's essentially showing such a pattern? I'll erase just so you could see it as well. Yes. In what? Yes. So, well, it's similar in that regard that essentially you have a finite amount of space. Here, you're constraining the amount such that this is how as much like you get smears of bacterium here following incubation overnight at 37 degrees. And then you repeat that process, but you're only taking a small amount, not like the amount that you started off with. So bear that in mind, everyone. Please pay very close attention that you're following instructions because you don't want to be making this mistake of going back into your NA slant with M. luteus and then repeating the streaking of your second quadrant. Rather, you're just doing it from the lower region of your previous quadrant and you continue going until you're diluting it all the way to the third quadrant, which I know looks odd as a shape, but the idea is if you were to look back, and this was your, let's say this was your plate, essentially, that's how your quadrats will look like. Essentially, post incubation. Actually, it's better though. Like this. That's hard to see, but hopefully get the idea. Like this, invert, or you use the round table, and then you go from the lower region of where you stop, then continue that process. And then hypothetically, if you turn it again, you would return to that location, or sometimes you do it from the outer edge, which is actually better. And then you repeat the process. And then post incubation, you'll have something similar to panel C. I'm gonna just give the slides. Make sure that you're able to answer this question if you haven't shown me earlier from the slides. Actually, let's okay, I'll continue. Serial dilutions. So just bear this in mind. Once we're done, we could go back, but I just want to go through all the slides so that way you'll have more time. I mentioned this earlier for serial dilutions. You're looking back into the past, right, on your different plates that you've done, the 3 plates with the different dilutions we've talked about. Now, for spread plating, you know it's different because essentially what you were doing was using the L rod to spread it around like an even cake or a pottery, right? Whereas in this case, you're not doing any direct dilutions within a single plate, but rather you're just doing serial dilutions from last week and then seeing the results today. But essentially, the idea is that when you look at your colonies, they should be between 30 to 300 per plate. If you find that's too much or too little, then you could essentially make a note that there are too many, essentially too many to count or too little to count, as shown also in the manual. But the idea is that when you, let's say, get your colonies, let's say we're looking at this, let's say we're looking at the second plate shown there, has a dilution factor of 1 to 1,000. And something to keep in mind is that if you look here, the original inoculum, right, from the top test tube, which is invisible, but essentially the tube on the right of it has 1 mil of bacterium within 9 mils of solution. And that would essentially lead to a 1 to 10 dilution, right? Which is why you have 1 to 10 essentially here at the top. The second tube is 1 to 10 out of that, the previous tube, so that makes it 1 to 100. But then because essentially, and that would continue going forward, but as you go down, instead of taking 1 mil into your plates, which would be a lot, you're doing 0.1 mils, and that signifies that you're doing an additional dilution. And that's where you get not only, like in this case, 1 to 10, but you get on your plate 1 to 100. And then in this case, where you have a dilution factor of 1 to 100, you go down, divide by 10 again, and that's 1 to 1,000. This is important because let's say, as you look back into your bacterial counts or into your agar plates, if let's say you get 35 colonies, then what that means is you essentially would do the inverse And what you're doing is multiplying 1,000 by 35. So you would have in total 35,000 CFUs. They have different units. Essentially cells per mil. And that's an estimate, right? Based on what you can actually see. Some cells look like they're, undergoing binary fission. Other cells look like they're just quite large as colonies. So just bear that in mind. But nonetheless, that would be your calculation: 35,000 CFUs or cells per mil. So yeah. So bear that in mind. That could also help you answer this question if you haven't done so already. Some questions are straightforward to answer in your lab notebook. Others may take some time, but this one hopefully you'll be able to do with ease using the same analysis as before. Now the plaque assay is on your benches. The materials include a lambda, right? That's your lambda page, and E. coli, which you also have here. I just want everyone to pay close attention. Besides the plaque assay, whenever you're dealing with bacterium, make sure once again you resuspend. Continually make sure that you resuspend your bacterium, so that way, especially in conjugation, you don't run the risk of essentially having minimal bacteria to work with. And similar to this in the plaque assay, even though you have the E. coli suspended, you just want to make sure that they maintain their presence throughout the entire supernatant or liquid. And now, essentially, apart from what you've done before, this step is a bit different in the sense that you're making, if I'm not mistaken, controls and your experimental treatment groups, wherein you have, like in the case here, 100 mils of water, DH2O, which should be on your benches, and the phage, which I said earlier is in lambda. You also have, based on your apparatus, separate, but for a transformation. But the idea is that you have the materials that you need from present in the lab notebook, and pretty much what you'll be using for today. Essentially, imagine what you did for last week's serial dilutions, but now you're adding in E. coli. Adding. Yep, you're adding in either E. coli or the phage. E. coli or water into tubes wherein essentially they'll be added into these agar, soft agar plates, tubes. After that, essentially, you place them using spread plating onto these plates. You have the NYZ plates present on your benches, but we'll talk about that when permissible. Lastly, this is your transformation assay. Oh, sorry, no, transformation is now. Let me just see the conjugation. Yeah, it's at the end. Okay. So the last page, if I'm not mistaken, is conjugation. So then, let's see time. Okay, I could go through this briefly then. Transformation, essentially, the idea is that you're going to be, inserting a gene of interest into your plasmid, right? And so there are different treatments and different conditions that you have. You have the ice bucket that I mentioned earlier that will help with calcium chloride, and and so bear that in mind. Essentially, that'll help essentially insert your plasmid of interest into your bacterium to permeabilize the cell wall. And eventually you'll have these cells grown and hopefully visualized. Because the idea is after incubation, after incubation at 37 degrees, you'll essentially have these plates wherein colonies would grow. And if indeed they have essentially the entire complete plasmid vector, then you will hopefully expect to see GFP. GFP fluoresces with UV light. So in a week's time, if we're able to see successful transformed colonies, right, of E. coli, then hopefully you should be able to see under the UV light green colonies. So that also helps understand what you would expect to see here. So just bear in mind the E. coli is down there. I mentioned it earlier, but you have set up the conjugation here. You'll have set up the luteus For sheep plating, exercise 1. That's exercise 3. And then for your, including the plasmid and Cl or calcium chloride on your ice buckets as groups, you have the materials that you need. And then conjugation is also on your benches. Once again, I just want to reiterate this. Please make sure that you resuspend your bacterium. And actually, we're going to begin essentially with street plating and then go straight ahead. So that'll probably be 10-ish minutes at max. And then bacterial conjugation will be the next exercise because we need to let them essentially mix and incubate for 2 hours. Then we'll be essentially spreading them on your agar plates. There are different agar plates essentially based on the different bacterium that you have, and they'll be useful to essentially showcase if the bacterium have conjugated, i.e. the plasmids that they have that support resistance in one bacterial colony over another. If they perform well in conjugation, then you should hopefully see colonies present in your doubly antibiotic treated plate, which is chloramphenicol and rifampicin or rifampicin. Yeah. And then you essentially it's very simple, but we have to actually spend a good amount of time on this exercise. So I would say let's go straight to street plating now. Once again, you have the materials, and just make sure that you clean your bench. And remember aseptic technique. Thank you. Two is very important that you're not making more than you need, especially for the use of the week other. You can essentially just plating with 1 to 2 touches on the end of your previous screen. You just bear that in mind. If you land this as you up three, I would recommend that as line green. Yeah. Around. On their started at its edge. Make sure as well when you're looking at your agar plate that they're close by to your Bunsen burner because that's part of the aseptic technique. You don't want to run the risk of cross contamination. Did anyone take more NA plate than needed? For street plating, you only need one. So please make sure if you're taking more than one to return it back to the middle bench. Sorry. Has anyone taken, question for everyone, has anyone taken more any agar plates than they need, like more than one for street plating? Because we should have enough for everyone. So please, if you've taken more for your bench, please return them to the middle bench. Just make sure that you label them on the edges so that way you're able to note the different morphologies that you see in a week. And also, if anyone hasn't shown me their lab notebooks, please do so as soon as you can. Okay. Yes. Where do you place it? I'm gonna place it in the incubator. In the meantime, with your bench, when your bench is done, tape them up, and then that'll make it easier. You can just put a label for your group saying, you shouldn't be. Yeah, down there, right beside the ethanol. You could just say your group's label with the practical number. And then if you could add in exercise 1, that way separate them. Thank you. Oh, no. By the way, if your group is done, please tape all of your agar plates, 3 to 4 of them together. And that way we can place them into the incubator as groups all together. Bear in mind that you place your group's practical number, individually your names, of course, but in your groups for the street plates, place the practical number, number 2. Your group number, I mentioned A, B, C, D, E, F before, and the exercise, so exercise 1.
Underscore. That was just the data for submitting. Go through on the notebook as well.
In the thing, it's
Another point for everyone, just bear in mind in terms of your discussion for future reference of your lab manual and even for the unknown assignment. For your assignments, please make sure that you include your discussion points inside your in your notebook as well. That includes the unknown assignment, the FLR, and any other upcoming assignments. So that way you keep your lab notebook as complete as possible. Thanks. So in the next, at max 7 minutes, you should be done exercise 1. And then let me know. So that way, once again, you tape as a group the exercise, your group number and practical. If your group is done exercise 1, you can make way to exercise 4. Just let me know. When you're ready, you can go straight ahead to exercise 4. Place them all together. All together. So when your group is done, you can start placing the plates inside, like all together. Wrap them with tape. We have tape in the middle bench for you to use, and you can label it even on the tape itself, and then put it out with your bench, your group, your practical, and anything you need. Does anyone have an extra inoculating loop with them? Yes.
It's the 5 milliliter we're mixing up.
For conjugation? We label it with your bench. All the details, bench number, practical number, actually, yeah, bench number, practical number suffice, and the label of CF, the two bacterial, HT90 and RFLI. Yes. Do you have someone who's online who would like to? When your group is done, exercise 1, please bring them here on this side, and then I can take them into the incubator. You could tape them and then label them like this with the exercise, the practical, and then. When you're done, you can place back the amniotus to the red tray there. Please also don't forget for transformation, when we reach that step, you return back the 6 agar plates with HB101 full line.
So
When your group is done, also a good reminder for what you do with grace. Don't forget when you have your pipette tips open, because we also want to maintain a septic technique that they're close by to the ventilator. Everyone has given me their plates for exercise 1. Please bring it in the next few seconds because that's what we need to see right now. So when you're done, let me know, and then we can all together move forward with conjugation. If you're already moved on, just make sure you're using the right materials because the white tray is the one that has what you need for conjugation, alternate tubes. HT99, J63R, and for conjugation. So, everyone else done? So next, I'm going to put time for 20 minutes to focus on conjugation. It shouldn't take too long. Okay. Thank you so much. Just to confirm, you guys suspended both bacterials, right? Yes. Perfect. Okay, so you can place the skill in your middle bench, in that middle tray, and then I'll let everyone else complete it, and then we'll put them all together. Oh, sorry, you're done, right? Yeah, perfect. I'll take them. No problem. You're done as well? Okay. Perfect. So just before, thank you so much. Now I'm just gonna go back here. Alhamdulillah, we've completed exercise 1. We're moving to exercise 4, as I mentioned earlier, because we want to move straight ahead for incubation. 1 to 2 hours is good. So just bear in mind that you look at the volume, 0.1 mils. You could use the pipette tips, the smaller ones that are in the purple or blue containers. So you have a P1000. Hopefully you all know how to use pipettes. If you don't, just let me know. But if you have a P1000, you could go to 010, that's 100 microliters or 0.1 mils. And similarly, 0.9 mils for J53R is 090. Could be added for and you need to spend your bacteria. And you suspend your bacteria. This is the common concern that some of us face. And I don't want you to get no result. Please bear in mind. If you find that occurring, just redo it with the samples that you have.
Okay, I need to ask.
I'll ask this one. No problem. So hopefully everyone's done exercise one. I'll be coming back a few things. Make sure when you're using the tips, your buttons are in close by. Make sure that you don't forget anything. So, how are we doing so far conjugation wise? I'll start the timer. So you have about 20-ish minutes. The sooner you're able to complete this stage, the better. It's very important that, remember folks, that we go with the flow of completing exercise 4 as a group. Make sure all members are paying attention, right? And then if needed, actually, another recommendation is having some of you focus on the conjugation and others focusing on transformation. So if you're able to separate into pairs, you can do this as well. However, I just want to make sure you're able to complete as much as you can as a group together before separating and making sure that everyone is paying attention and is fully efficient throughout today's practical. There is a question about using pipettes. How many of you are struggling to use a pipette so I can help both of you in tandem? Okay, let me just help this group and I'll come to you next. When your group is done for conjugation, let me know in about 3-ish minutes. And then conjugation, resuspending both bacterial H99 and J53R. And you take the right volume, 0.1 mil, H99, 9 mil, you follow exactly. Back to the upcoming group's questions. So there's a question about conjugation and how you're essentially going to use the sterile swab. It's shown here as a one-drop method. It's specified also in the manual. But the idea is for each bacterium, and please pay attention to this because it's different for all 3 plates. The first 2 are done the same with a one drop. Essentially, you've plunged your cotton swab into your bacterium. Make sure that they're resuspended, right? Once again, of your bacterium. I'm just looking for another marker. But the idea is you've added in one drop, like from your cotton swab. And let's say this is your tube. And it's sterile, right? Because it has the label with the hashes indicating that it's autoclaved. And so you've taken out one drop, you plunge it into your plate on one side, because bear in mind, you have still HT99 that you're working with and also J53R. So remember, for each condition, you're putting one drop and you're smearing it along. Kind of like what we've done with spread plating, but only using now a cotton swab. So that's what you're doing for the first 2 plates. because, and bear in mind they're labeled, right? So make sure you're using the right one. And now you're able to put a mark in between, so that way you could separate. This is HT99, this is J53R, that you've labeled and sliced your plate accordingly with a marker. And then essentially, you do this process for the first two plates. But then for your third plate, the one that's going to go into the incubator, that's where, as groups, you have to let me know when you're ready, and I'll place them inside. And then in an hour or two later, we're going to come back to complete that experiment, to finish off the third plate, which should be done in a similar manner. And then you're just smearing all along your plate. Just make sure, let's say you're done. Remember, in about an hour and a half, I don't want to forget this, so please don't forget as well. In an hour and a half, you're going to have a pellet at the bottom. So make sure, following conjugation, you resuspend in an hour and a half's time, roughly. When your group is done, please hand them the two screens, and I should be able to take them. When your group is done conjugation, let me know. At least for the tubes that need to go into the incubator. And you do that. When you're done?
Yes.
Either one, actually, please. We have one more group. Need to place their
Yes.
everything okay? What's your outstanding questions? I know some of you have different questions regarding Q as well. Yeah, so the reason I say this is because sometimes the degrees are not actually over the lab. So even that I'll talk to the lab, showing to their
Waiting. Already put the water and it's like full of all the food. Yeah, and we're just. The salty. Yep. I'm gonna say that's time. It's a
See, because there are different things that you could do. I just want to find out which one made more. Yes, the top agar ones are down there. I just want to exercise too. For the plaque assay, yes. So there is a soft agar that's there. Oh, I, I would have expected that you have. Do you have any saline tubes in rural trees? Generally small. I don't think. Yeah, I don't. Yeah, I don't think actually you could. You could use these ones. Yeah, yeah. So in terms of exercise two for the plaque assay, if the group is done, conjugation, then what you could do is make use of the TEPI tubes shown here, where the gentleman is taking them right now, the pen, and essentially we have extra ones here. We also have your slide box for those that want to see their unknown slides. Just let me know before you take them because some are on the very top. And in terms of other materials, yes, when you reach the stage of the plaque assay, when you're actually mixing in your E. coli phage mixtures into soft agar, we already have them here at 55 degrees. So for those that want to further, these are where your soft agar tubes are. You'll find them right here. They're at 55 degrees, a lot of them. Please bear in mind, once again, you label them, right? Because you're going to be using them for essentially to accordingly make those plates, the 5 different plates that you have with you for NYC. Um, for the control?
Like, is this that one's provided to us, right? The one that's boxed. So I'm just wondering.
You can. So let's see, download page with learning. Let me just make sure.
Is it on the lab video? Not on this side.
Yeah. Okay. Uh, just one minute. Yes, you have a question.
Please see a 37 degrees of the agar.
Yes.
Is that
You can place them into an incubator.
incubator?
Yes.
Okay.
For those that want to soften up their NYZ plates in the meantime, for conjugation, you can place them inside the incubator. Just make sure that you've labeled them because as groups, you want to be able to identify which one's yours and not take anyone else's. Sorry, there was a question about tube 2. Because technically you have the epi tubes where you can actually, so okay, you know how you're starting with phage, right?
Yeah, like, which one is the starting material?
You have the starting material with your lambda for conjugation, right? You're on this exercise, right?
Yeah.
Okay. So you have the tube that's labeled lambda, that's your phage. And so within that tube, you can add in, depending on the volume. Then from that tube, you're going to use other tubes to make the further pregnancy. So remember, epi tubes should be here. Please, once again, don't take more than you need because each group should have at max 10 epi tubes. Which, because we're working as groups, there should be enough for everyone. If the last group hasn't taken epi tubes, this is where you could grab them from here. There's also another thing. Sterile H2O is in the glass canister where it has a label of a hash, like this, just to indicate that it's sterile autoclave.
Yeah.
Anyone having questions so far? Any difficulty? Also, just to confirm, everyone's done placing, like making their plates for conjugation. Who's not done conjugation for their first two plates? Raise your hand. Group 4, you're done conjugation, right? For the plates, first two plates. Group 5, is your group done conjugation for the first two plates? Or sorry, the two plates for swabbing. Same for your group. And all three of you, I believe, as well. You're fine. That's the last one for about a week or two. So make sure you take it. And a pencil as well. Some of us have it on that side as well. If you're missing any belongings, please make sure to come and make sure you don't have anyone distracted. Yes, two, the one that shows page, the lambda symbol, that should be diluted. Now it's interesting because apart from that, what you will be doing is essentially making further dilutions. I would say tube 1 is the one that you're starting off with just water, even though it's at 100 microliters, this is step 3. So just bear in mind how you're essentially following the next tube steps. The ones that are tubes 1 or 2 to 5, those will have phages or lambda essentially diluted in the epitubes that you have here. 10 to the minus 3, 10 to the minus 4, 5, and 6. It should. Can I just see it one more? So for the upcoming 2 lambda, because essentially you're going to do it for tubes 2 to 5. It's important to note this because it goes down to place B to E, and that's where you're going to be able to make note of the different dilutions for how they impact your PFUs, which is similar to what I've talked about earlier for your original cell densities, except now we're dealing with plaques or essentially bacteriophages that essentially lyse your bacterium. So instead of what you normally see for your agar plates, in a week's time, you should hopefully see empty dots that signify the number of PFUs or plaque forming units. Because of the dilution series, serial dilution, following your plaque inhibition or incubation rather. Yes. Saltiger is inside with the pot. Yeah, that's it. So just for an update, everyone, we're halfway into the lab, but I want to get an idea. Where is everyone at? Group one. Are you at exercise 2, exercise 3, exercise 4? 2. And so just to confirm, you finished the first 2 plates, right? Yeah. Okay, so exercise 2, right? Okay. And likewise, group 2, are you on exercise 2? Plaque acid. Group 6, plaque acid. You guys there? Plaque acid. Group 4, Ferraro, Joseph. Is your group on plaque acid or another? Okay. So if you guys want, you could separate as pairs. Some of you focusing on the plaque assay and others on transformation. And then in an hour or so, we should be able to get back on track to complete the rest of the practical. You can stay here. I just want to make note of that. So as a group, please try to complete the plaque assay at least up to step 4 in the next 20 minutes. By 11, you should be able to pour in the NYZ plates. Please make sure that you're preheating your NYZ plates. Who hasn't preheated them into the 37 degrees incubator? Please do so now. Please incubate them now. This is something that's important for essentially maintaining the incubation period and condition for your bacterium. Group 4, you've incubated, preheated your plates. Yeah. Group 5, the same. Group 6, group 2. Okay, can you place them now? Essentially, the 3 plates. You just place them. Do they have bacteria or no? So you can place them inside the incubator. Just make sure they're labeled so you can grab them in about. Group 1, don't forget to preheat your plates. Okay, your question.
And then it says water to the control scene and acceptably transfer to the corresponding micrococcus. I don't understand, like over here, are we also adding the bacterial phase into these viruses? Is it just the E. coli?
So okay, you're pipetting E. coli, and actually that goes well with. So these are the 5 tubes. You're adding in 300 microliters in each of them, and then your question is.
Like then he said, the bacteria is out of the phase so does that mean now? Or is it just saying that?
It's just saying that because the next step is where you're actually adding it. So you added 100 microliters into the control only, but bear in mind that the 100 mils for the phage, yeah. So it's just preparing you for the next second other step. No problem. Any other questions? Remember, you're following aseptic technique with your tubes that you flame them. Remember at the neck that you flame your tubes to maintain aseptic technique. This countdown should help you all complete up to step 4, bearing in mind that you poured in, you've preheated your NYZ plates. These are nutrient agar plates that have different elements, metabolites present in them. You want to come if that's okay? The reason I want to make sure everyone's on the same page is also shown in your manual. But bear in mind, you're adding 300 microliters of E. coli on all 5 tubes, tubes 1 to 5. One is your control, where you're adding 100 microliters of water, sterile water, no phage. And then the rest of the other ones, 2 to 5, are where your phage and then the diluted or the dilutions of your phage are added in. How come?
So it's 23 seconds, right? As we are adding it, it's already finishing 3 tubes. It's not going in 5.
How much did you have to start?
It's 4 pages filling.
So that would have been 1.5. You should have enough.
Should I be doing a wrong calculation?
One minute. Okay. How many groups need more E. coli research on? 1, 2. Okay. How many of you have completed all 5 tubes with enough E. coli? 300 microliters. 1, 2. 2. How many? Let me see. Okay, thanks a lot. I just need to calculate. Did you complete an entry about what? Have you done all? Oh, the last test is not really fun. I know some of you may be waiting for your E. coli, but continue on with the remaining tubes. The tubes that you have already prepped, you can go straight ahead for the 15 minutes in the place for incubation or for the phase If you need rats, place it. If you wanna place your tubes For preheating into the incubator, we could use these extra sets, these racks. So just bear in mind that you let me know. 42 degrees, we're using this water bag, 55 for the soft egg or
I need one.
I think there's yeah. How's it going?
I think we did it.
So, for those of you, attention everyone. I know some of you have been asking about, and you have about a minute to reach step 4 for the plaque assay. I just want to make sure that everyone's on the same page though. For the E. coli, technically I spoke to the lab tech, there should be enough. But to make it easier for you, see how much you've added in the microtubes thus far. There should be 300 microliters of E. coli. Some of you have actually added more because you may have actually gone to the second stop when you're pipetting inwards rather than outwards. Outwards, you go down to the second stop, but inwards, you're only taking it from the first stop. You don't press it all the way to the bottom. Otherwise, you're taking over 300 microliters, and that's why you may not have enough for your other tubes. So if that's the case, what I recommend is with me, essentially having a look at how much you've added in, try to subtract that amount. It'll probably be 150 microliters for the first two or second set, like this tube number 2 and tube 3, that you're going to be taking out essentially a finite amount to place into your last set of tubes, because you want to make sure that you have enough, and then hopefully that should account for any lost E. coli. Anyone else having problems regarding this step?
Is this the equivalent
For transformation, right? You're asking for transformation? It should be. Yeah. Yeah. The piece for the plaque. Yeah. I placed racks inside. I'll give you an extra one.
You have a tiny bit of.
I would say it's pretty okay, but what about your last? You can go. When they add it, how's everyone doing?
That's your right floor. I was holding 14. Your hands are. Your hands are.
So some of you have been asking about exercise 3. Can I just confirm if all of you are done exercise 2 with the plaque assay? You're done? Your group almost done or done? Okay, okay. So about 15 minutes, I'm guessing. That group, okay. Your group is almost done. Conjugation, plaque assay. Okay. So you've already you're heating them up. Like, are they now in the incubator? Aman, are they into the incubator or no? All of them? With the colonies and plaque? Okay. Group 5, are you guys done exercise 2? Yep. With the bacteria as well? The bacterium and plaque, like the bacteriophage as well, like the lambda, like everything. Like, have you reached step 5? They're heating. Okay. Okay. From the transformation plate.
To like describe this from last week's.
Yes. Does anyone have extra E. coli? If your group has extra E. coli, please raise your hands. I would just go with any remaining, and then it should be okay. If any other groups have additional E. coli for exercise 2, please give it to group 6. Thanks a lot. Now, yes, that should be okay. That should be okay. Now let's see. Bear in mind, please don't use your phone. Okay, but you're not using it for your picture right now, are you? You know the reason I'm saying this because I appreciate you're ahead of the game, but if Dr. Brunk comes, I don't want you to get in trouble. That's why I'm saying it. So just bear that in mind. So you have as a group under 15 minutes, actually we'll say 13 minutes, to complete this final stage of the plaque assay. So likely most of you, oh, by the way, what did you put inside there? Nothing? Okay, you opened it, okay. So step 5, right, just to make sure everyone's on the same page, right? You would have taken the soft agar for step 4 in there. That's where this core is taking the soft agar, the 5 of them, if I'm not mistaken. Label them once again, right? Because following this, and remember, resuspension of your tubes with the phage and bacterium. Thereafter, you're going to be pouring them, reminding yourself of aseptic technique, right, in the NYZ plates that you preheated in the 37-degree incubator. You can make note of these methods also in your lab notebook. They may help and be beneficial. And in the meantime, if your group has completed this stage, this is your time to look at your serial dilutions from last week. You can start counting the colonies. So remember, if you've had in each plate for the serial dilution last week from the spread plating technique, if you have colonies that are 30 to 300, essentially after counting, then that's where you essentially do the calculation. I showed it before. You're multiplying it by the final dilution factor, i.e., if your tube was at 1 to the 100, essentially 1 divided by 100 dilution, and you added in 0.1 mils into your plate, that's another time 10 dilution, meaning that you're going to 1 to 1,000, meaning that your final dilution factor is 1,000, 1 to the 1,000. Thousandth, and essentially you're multiplying 1,000 by the number of CFUs or colonies, individual colonies you find in your plate, and that should give you your original cell density for 2, if not all 3 plates. Try to do it for all 3 plates, record the results, and find a mean or average as to what all 3 would be to determine your final or expected, observed original cell density. Hopefully that makes sense for everyone, or mean cell density. You could use a marker to count the dots. You can also use your mind as well. I like to count with my mind, but you can also count with a Sharpie as well. So you have 3 plates, right? If it's too small or too large, that's okay. Yeah. So you just focus on the other 2.
Yeah.
Yes. Yes. We Thanks a lot. So in about 20 minutes, we're gonna take them out, and ideally, actually 20 minutes, yeah. So 20 minutes from now, they're gonna likely form a pellet. So just like how we did it before for the other bacterial species, HT99, J53R, we have to resuspend them because they're now ideally supporting conjugation between both bacterium. So we want to make sure that we get them all in that resuspension and not at the end of the pellet for spread plating. Or sorry, spread plating with the swab. No problem. When we're in about 20 minutes time into the conjugation final plate, bear in mind that you're following the same procedure as before. You're resuspending your bacteria. Remember everyone, resuspending bacterium always. And follow aseptic technique. You flame your lids, the neck of your tubes. Make sure that when you're taking, 9, I believe did it specify? The amount with your swab, just make sure that you're following aseptic technique as you take in and out your bacteria, and that you, of course, resuspend well. What are you looking for transformation?
Yeah. It says after 10 minutes of iris transfer to using micrococcus folder. Can we just put it directly to this side?
There is, and just to confirm, it says 42 degrees. Yes, so you can. Now the other thing is you want to also complete the number of CDs, original cell density.
Oh, we already did that.
Perfect. So just make sure that you record the OCDs, the essentially original cell densities for your spread plating last week from the serial dilutions. That'll be part of your post lab for the results of this week. If you have over 300, you likely have 2 other plates that are within 30 to 300 colonies. Use those plates to multiply by the dilution factor, the final one, of your plate to determine the original cell density. Yes, it is. Well, actually, which one?
Like here, 250 LB
Yes. No. We're good. I spoke to him as well.
Okay.
Thanks a lot, actually. I really appreciate it. Everything else should be okay. That's good. That's good.
I'm just gonna blah, blah, blah
For the plaque assay or?
everything is like they're just incubating.
That's,
So
yeah.
they're just incubating and I started the other one first, the conjugation, because that's like
Yeah.
2 hours. It won't be done till like 40 or whatever 40.
Yeah.
So
This should be okay. Technically, manual says 1 hour. So
oh really?
yeah. How about,
I did not do that.
let me show you.
I did not do that. Really? It's a good cheat.
1 hour. It's okay. It's okay.
I didn't see it.
If even if it's like 10 minutes more or 30 minutes or it's okay. Oh, by the way, one thing they told us, don't forget to resuspend. They need to
I
resuspend.
know. I told them like that because it's.
Yeah.
Okay, we'll see you with the data next week.
All the best. No problem. Yeah. Okay.
That's what we're trying to figure out. We're like, oh, why is it's noise. It's like it's all in the appendix, so it's like scroll over here. It's like they have like little.
Yeah. Yeah.
I'm gonna go back over there. Okay.
Yeah. When groups are done for transformation, please make sure you return the plates back to where they were originally. Also, when you're taking the colonies, take one, two small ones, one big one is enough. Remember, aseptic technique when you're using the plates. Pipette tips, if they're open, there should be a button on as well. I can see with this one, the box should be closed. Where? Where is? How's everyone doing so far? Good? Yes. So, kind reminder, we have, well, it says 47 minutes, but we are in good timing to almost do the conjugation for the third plate for this final exercise. So I just want to make sure that before we end off, please bear in mind the following. So we've talked about transformation briefly, right? And I mentioned about the GFP protein, right? Following successful conjugation, the colonies next week should be able to fluoresce in UV light as an indication of successful transformation with GFP and the other regulatory proteins. So just bear this in mind. It's similar to what you've done in B12. So bear in mind, everyone, that once again, you're following what we've done before, and you know. No. You said no, right? You said it was hardened.
No. Yeah, you do one at a time. And now those are lost. They are not going to, they can't actually come solution again, and if they were, you would have to put them in. You only take one of the red cap tubes out at a time. Okay? So don't take the last one out until you're absolutely ready to add the bacteria, right?
So just want to also, okay, so as I mentioned earlier, right? Remember, based on what we've talked about, for the conjugated plasmid, that's going to fluoresce green next week, right? But make sure that you've done this correctly. And I also want to highlight what Dr. Brunt said, because it's important. Everyone, we're paying attention. Everyone's paying attention. Okay, so please bear in mind, some of you may have tubes that have hardened. So bear in mind that if that's the case, we have two options. One is reheating, though ideally it may actually run the risk of denaturing the proteins of your bacteria. So what I would recommend is redoing for those tubes. I would say it's 3 of them for some of you, redoing that step with the other tubes. I know, though, it may be the case that you don't have extra. Actually, you should have enough. You should have enough to complete the rest, but just bear in mind that you're following instructions, because one at a time, and this is something I forgot to mention, but one at a time, you'll be completing these stages for the plates. So bear in mind that you're following the instructions, soft agar plates. You've added in 0.4 milliliters of your E. coli with your phage, or diluted like DH2O. So essentially just follow the steps and you should be successful in completing the rest of today's lab. And likely in the next 2 minutes, all of the NYZ plates should be ready for incubation overnight. So let me just count with everyone. Group 4, are you done the plaque assay? 2 more minutes? Is that okay? Okay, how long for you guys? You done? Group 6. Group 6. How long? 2 more minutes, okay. To complete it for the NYZ, please. Okay, so I need 2 people from your group to go to the final conjugation. Transformation or conjugation? So how long until you're done, both of you? So I would say at max, I would say around so because you don't have enough time, you're gonna have to go to the conjugation. I would recommend in 7 minutes that you should be done. One of the pairs of you should be done your exercise, and then they should go to the conjugation for the last plate. Yeah. So the plates themselves are there. So for so remember, everyone, aseptic technique, especially when it comes to the line, we want to make sure that once it's returned back, we're able to complete and make sure that we're doing everything else right. So we have, I think we should be ready, hopefully, to complete the 3rd plate for conjugation about now. So I would just say for group members, now is the time to start splitting up to see who's going to be able to, instead of 2, complete the conjugation experiment. Remember, aseptic technique, you're flaming with a Bunsen burner, your area as well, 70% ethanol, and make sure that when you're doing this last plate, you resuspend your bacteria, very important. Resuspend and then take out 1 swab and then spread across the entire plate. Make sure that you do this properly, everyone. Otherwise, if you don't see results, it may be that you didn't resuspend correctly. Any questions? Can you just label?
Take care of the colonies away from right there anyway. I see.
So the incubator, you're placing it on the top left side. We don't have actually all locations on in the incubator, so please bear that in mind. You don't want it to be mistaken for another group. If you placed it in the bottom left side, please place it up towards the top left because we don't have access to the other locations, and you don't want yours to be mistaken for another lab.
Are they getting it out already?
Right here, but it's not. For the. No, this is the other. 95. Yes.
That increases the likelihood of getting sick. This is.
You're doing going.
Everything okay for you? Remember aseptic technique. Yeah. Transformation, not seen at all. Now, there's a lot of classes. Some of them are empty. A good number of them.
Not standing where it was sitting.
So now everyone should be ready to do their last plate for conjugation. Remember, for your third plate, before you begin, remember aseptic technique. You're flaming your tubes, but also don't forget you suspended the bacterium, remember? Because we don't want to have the risk of them just being submerged as a pellet and then running the risk of not having enough to actually visualize for next week. Keep it up, everyone. Do your best and leave the rest with the best. So I don't want you to delay too much, but we have about half an hour. How many of you are doing their conjugation right now? Group 5. Group 4, are you done conjugation? You done all three? Conjugation, transformation, plaque assay? Like the third plate for conjugation, is that done as well? Or no? Okay, so complete it now. You should be able to take it out. Remember aseptic technique, everyone, for conjugation. Also that you resuspend your bacterium. Group 5, how many more exercises are left? Plaque assay, transformation, and conjugation. Okay, so can one set of you do the conjugation?
Yeah.
When your group is done, with all three plates, you can start them up, and then we'll take them back.
The broth, are we done with it? Do we dispose of it or?
You can place them back into your bed for exercise
No, for four, the like antibiotic slide things, plates. We put it on and then but I pulled up, so it's a lot. Yeah. Now literally. What is else? It's a hard one.
So if you guys are done, most other parts of today's exercise, remember the conjugation can be done now. I've set the timer and it's already off, right? So bear in mind that for the last 15 minutes, we need to make sure that we're done the conjugation. Very simple once again. Remember, you're resuspending your bacteria and that you make sure that you've labeled and added in enough smearing such that you're able to accurately complete the rest of today's lab successfully. When you're done, just remind yourself of what's next, the post-lab as well, the results that you've done for today for the counting of your previous fragments, and then any other upcoming for questions, John.
So next it will be LB and LB 200. Correct. Yeah. So this is gonna go with LB A and LB, and this is LB. Look. Yeah. Yeah. Oh yeah, I'm sorry, I'm looking at.
Guys, you know that looking to the count, it's like circulating, counting the number of colonies, you can place them back into autoclave bin. Down there, Q-tips, anything that touches bacterium, you can place it into the autoclave as well. Remember for your transformation assay, please bring back the E. coli. There should be 6 plates back here. HB101 labeled there on. So just make sure that you return them. Can I ask for a status, like how everyone's doing so far in terms of, I've seen a lot of you are cleaning up. Have all of you also completed, so just to confirm, we're done transformation, we're done the plaque assay, are we done conjugation though? Yeah? Okay. Remember that once again, 6 plates for transformation need to be returned back to the middle bench for use for future practicals. If as well you wanted to see your unknown slides from last week, you can have a look at the middle box found in the middle bench as well. Into the article. For the ones that have bacteria. Not those ones. They're going to be reused. So, okay. When you're done, place them back to the down there. Someone also had a red tube, a tube that perhaps has bacteria for the black assay. Anyone left it inside? Is everyone done all experiments, or is anyone almost done? Missing 1 or 2 more experiments? I just want to make sure though, done conjugation, done transformation, done the assays. Everything's labeled, all active dishes are back inside, or the agar plates, all of them are back inside the incubator. In terms of the remaining, okay, they're done. There is one other thing, post lab. So let's talk about this in the meantime, just so everyone is on the same page. Before you leave today, make sure that you answer the questions here. So with regards to the transformation lab, the different components that are referred to in terms of the plates themselves, one, because you're adding it into different plates that have either, like essentially would include ampicillin with your LB broth or with or without the DNA plasmid. So be able to answer these questions A to E for your post lab, as well for the calculation of your cell densities. That's something you also want to include from the serial dilutions from last week. As well, make sure that you can actually start beginning to prep for your formal lab report with the introduction, right? And results you'll see hopefully soon, but you need to make sure that you're ready and prepped for this in due time. Now, lastly, or let's see, before I end off, this entire bench is done. Let's just erase. Sure, your plates are also inverted. Some of them, hopefully all of them are inverted. And just make sure once you get your unknown lab assignment, if you have questions for marking, let me know. Hopefully all of you made our plates. And just remember your post lab includes the results from the week that you've been able to come up with from last week, as well as the discussion questions, those in the manual, and also don't forget your pre-lab for next week. Following next week is your reading week, so I would say start preparing for your lab exam, because you want to make sure that you're able to complete as much as you are able to from weeks 1 to 5 in understanding, and that way for the latter half of the term, you'll be able to more easily focus on the last set of exercises. Once again, lab report or lab exam is likely during your final exam. Yeah, but you want to prepare for it now. The sooner you can, the better. Also, when it comes to just preparing for the lab exam, make sure that you're able to understand the content that we've gone through, as well as questions that may be asked about what happens if you made X, Y, Z mistake, or what happens if a colleague finds a new type of protein, and how you'll be able to uncover certain mechanisms behind them in a bacterial assay, or likewise for staining techniques. So bear all those different details in mind before you leave today. With that, have a great day. Take care, everyone. Wishing you all the best. And if you have questions for your unknown assignment, I have them marked. If you have questions, let me know. And I have scaled the grade 10% because there have been some concerns that I do want hopefully in due time to talk about. Just make sure that you do your best to complete as much detail as you can from the observation to your flow chart and making sure that you have nice quality results. Not just the slides, but for upcoming exercises and especially for your formal lab report with greatly detailed figures and results. So with that, take care everyone. Have an amazing day. So if anyone left their contact lenses inside this tray, this is your consultation. Some of you may also be missing some things that are important, but I don't want anyone leaving the lab and losing anything that they need. No problem. Make sure that you always take what belongs to you, not to anyone else as well. Make sure that, especially when you have things that are on your bench tops, that you spray ethanol. So that way you don't cross-contaminate your phone or your pencil cases or any of your belongings. If any of you have questions regarding your formal lab, I'm just going to answer them. So to help prepare you guys, if you come up with questions and want me to have a glimpse of your lab report 3 days in advance, I'll have a look. I'll give you feedback. And that way you can prepare for it better for the actual assessment, the grading of it itself. You also have the rubric and the details also posted on Perkins, hopefully. But if not, at least you can start asking me questions now. The sooner the better. And I just would kindly ask, try not to ask questions like the Monday evening of our practical because I get a lot of emails and I don't want to miss out on your questions, but also bear in mind that I'm seeing for the bit difficult. So it's just to bear that in mind for practical rather, 2 courses, but essentially just bear in mind that you do things efficiently. You're done quite early in advance, so with that, take care. Have an amazing day. If you have questions, let me know. If you have anything also like stains on you in future use, feel free to use this as well. Take care. Everyone have a great day. Likewise, have an amazing day, all of you. Make sure that your plates go up to the top, by the way. I see some are at the bottom. Unless you're just sorting them now, I just want to make sure that we don't keep the wrong ones. Thank you so much. We're,
Yeah, the bio AO2. I applied for it last semester and I got it, but I rejected it because I was so scared I wouldn't be able to handle it. Do you like it?
It's okay. Well, I'm a TA, so it's different for students.
You know, your master's in public or pursued doctorate, right?
I actually graduated, so
Okay, that's good, because I wanted to do it, but I was courses, 2 labs, tutorials, two tutorials. I didn't think I could do it, so I rejected it, but I really wanted to. I wanted to see if how I did on the Alaykum assignment.
So give me 1 moment. I'm just gonna make sure that everything is good. Would you be able to see it around 12:05? Is that okay? Or if you have a class, then is that
Sounds
okay?
good. I was gonna go crazy. We don't have.
Okay. Is that okay though? Or like if you have to go, it's okay. Yeah. Like for the or budget. It's okay. And then if you want to ask me via email, it's okay.
If you can, are you gonna freeze over?
Yeah, but it's okay.
But if you get, if you get a chance to make my grade, if you don't mind, you can just tell me later.
Email would be better in case of no problem, no problem. Anything okay? That's good. Three.
I'm 20.